Good morning, everyone. I'm welcome you all to this 21st day of CSIR online question and answer discussion session. Today we are going to discuss about the Unit 8. Okay, wow. In this Unit 8, we are that the topic is inheritance biology. As per our schedule, we are discussed the unit previous units 4, 5, 13, and then 1, 2, 3. 6 7. Now we are going to discuss the unit 8 because we are, in this session is fully going to discuss about the part C questions of the inheritance biology. It's just a glimpse of the unit 8 because we are not cover all the questions because we are in a short term of time duration. So in this session we are going to cover the little small questions. Okay, we will enter into the session. Okay, the total variance in a phenotypic character can be split into two component genetics. That genetic component is known as the VG, and the environmental component, that is VE. Okay, the genetic component, that genetic variance, that is the denoted as VG. The environmental variance is denoted as VE. In the inheritance of phenotypic trait can be expressed quantitatively as heritability coefficient H square. The heritability coefficient is H square, which is calculated equal to, the equation is the C1 Vg, that is variance depends on genetics, divided by Vg plus Ve. The C is the answer. Okay, Vg means that is the genetic variance. Ve means that is this equation Vg by Vg plus Ve H square, that is the heritability coefficient. Okay, next question. In neurospora, a cross between the genome A and small a result in an ascus with the ascospore of genotype as shown below. Ascus having the genotypic the arrangement. Okay, once again I repeat, the neurospora, that is having the cross between genotypes capital A and small a, results in an ascus with the ascospore of genotype. In this type of question, statement of P to S are event that could have a occurrence during the meiosis. What happened in the meiosis? They are asking the what happened in the meiosis. In this kind of question, we want to know the basics. Then only we can easily answer. So just neurospora. Consider of this unit 8, you want to study some important topics of neurospora, then what is genotype, what is ascus. The genotype questions are very repeatedly asked in the CSIR question. So just go with the genotypical quiz, just attend the quizzes and get better knowledge. Okay, first statement, P to S event could be occurred during the meiosis. What they asked, the above event could correctly explain the observation between shown the figure. What is the they are given a P, Q, R, S. In that, which one is the correct explanation for the particular figure? They asked. First thing, the answer is R. Answer is the A option. A is the P, crossing over between centromere and the gene, and then segregation of allele A, capital A and capital small a in a meiosis 2 phase. Okay, what happened? Crossing over the between the centromere and the gene. Centromere and gene are crossed over. Then second step is the segregation of allele meiosis 2. Okay, next one is it's very easiest question. This kind of questions are they are given a story. Okay, the story is fully covered the entire session, entire content of your answer. So just to follow the question, the following is for the development of white type red eye color in an insect. The red eye insect, they are having a hypothetical pathway. for red eye insect. What is the pathway? W, that is colorless precursor that react with enzyme A and make a orange pigment, that is X. Then colorless precursor Y, that is react with enzyme B pigment. This orange and brown pigment are mixed together and they are make a red eye color. Okay, this is the reason for that red eye. So what they asked here means the enzyme cap A and B is encoded by gene A plus and B minus respectively. The following statements are made together regarding the inheritance of a gene involved in the development of eye color. So as per their question, we want to understand. Sorry. Okay. As per their question, we want to understand which statement is correct regarding the inheritance of gene. Okay, the correct one is D, that is the D option, PQRS. Many of the CSIR questions are look like this. They are provided the four statement. In that, they are asking the which one is correct statement, which of the statements are correct statement. In this, most of the time, the combination of statements alone, PQ or PS or PQRS, this type of combinations alone be an answer. So whenever you are answering the CSIR question, just remind that if we are find any sequential question, this kind of statement question, just read out the options and question. In that question itself, you can get the answer, because that most of the CSIR exam questions are like that only. They are having a group of information in the question, and then you can get the information from the question also. So please go through the question well and then get an answer. So here question is they provided this pathway and then they provided this. As per their statements, P is, what is the P? PQS is the answer. R is the wrong one. PQS is the right one. What is the P? When two heterozygous individuals of phenotypes A and AB plus B, this are mated progenies with red, orange, brown, and white eye color being observed irrespective of whether the genes are independently asserting or showing an incomplete linkage. This is the one perfect match statement for the pathway. Next one is when two heterogeneous individuals are phenotypes A plus AB plus B are mated progenies with a red, orange, brown, and white color eye. They are observed ratios. Okay, they are irrespective of whether the genes. Here they shown are respectives. Their genes are having some ratio. N 3 is to 1. When genes are independently assorting. Okay, they are complete linkages. Next one is when a heterogeneous individuals are of a genotype A plus B or A B plus is rest crossed. This is a different one. It's occurred in a genotyping. Progenesis with orange and brown eye with more in number. This is the this is the reason. These three statements are the two one. Next one is easiest question. The Mendel cross tall plant with a draft one. Draft 1, the F1 plant were all tall. When these F1 plants were selfed to produce F2 generation, they are produced F2 generation. He got 3:1 tall RAF ratio in the offspring. They are getting a 3:1 ratio after the F2 generation. What is the probability that our three plants that in a F2 generation picked out random at a random, 2 would be a draft and 1 would be a tall. What is the ratio? Actually, what is the output? At the first generation, there are Mendel crossed the two pipelines. The first generation, they are crossed. They may get a F1 plant that are all tall. For that obtained 4 plants or 2 plants are tall, and the F1 plants are selfed. Okay, selfed to produce. Self means they are not cross-linked; they are directly produced. That so how the in that F2 generation he got 3 draft and 3 tall and 1 draft ratio in a offspring. What they ask means they want to ask the probability of 3 plants in a F2 generation become if they are taking a random means that want to be in a draft, and next one is that should be the one should be a tall. What is the probability? Okay, here they want to do the experiment, the mathematical modeling. That modeling that you can get answer 9/64, because that's first the probability of the getting plants is more than, because 4 into 4. Okay, that will first step the F1 plants are 4, and second step also that is a 4, the third step also 4. So this is the probabilities of getting the getting probability calculation. So in the first step they mention he got only 1 draft. Okay, then he want to do the multiple experiments, multiple generations. So that calculation gives a 9/64. Then next question, affected individuals from the pedigree given below of suffering from albinism and autosomal recessive disease. Identify the confirmed carrier individuals in this pedigree, assuming that members coming from outside that families are homologous or for the dominant alleles. They are homologous for the dominant alleles. This is the statement. They provided the story. Now they ask first generation. This is the normally the square box is mentioned the male, the round circle is mentioned the female. The next generation there is no dominance, though no, there is no mutated, no deceased one. So in the 4th generation only they have found the deceased one. What they asked in the question, they identify the confirmed carrier individual in the pedigree. Assume that the members coming from the outside of the family that are homologous. Outside means you can find here this in this first generation this person. Okay, the round is from the family, the square is from the out of the family, and in this second he may be the daughter. A daughter married a outside person. Here the son married the outside person, and in this step the daughter married. Okay. In this point of view, they asked what is the expected outcome. Outside person may be the homologous person. So about this question, first C is the answer. Here they asked the third generation, third generation second person. What is the third generation? Here this is the third generation. That is the son of the first generation mom. This person may got the dominant allele, because this is a first person, outside person, he involved in this person. The second one is the third generation third person. Third generation third person is here, and third generation fifth person here. Here this fifth person allele of this second generation mother or external mother. Okay, next one is third generation 6. This person is having this outer side person. Okay, this is 6 from 6 they are made. So this is the outside person. Second generation to lead from this disease, this only we are moving this side. Next one is second generation, second person. Okay, second generation, second person. He's affected. He is dominant allele because that is the internal family. They are affected with the external family. Then second generation is a family, internal family. Then second generation, fifth person. Okay, just we want to understand who are all the they are dominant. Okay, coming from the outside the family, they are homologous for the dominant allele. They are make a involvement, higher involvement. Next, just think about this. Assume a 1:1 ratio. What is the probability that three children from a same parent will consist of two daughters, one son? What is the ratio? The ratio here, the 1 by 3, that is the 0.37. Next one is consider a following cross involving gray wild where is gray involving gray, that is wild type, and yellow body color, true breeding Drosophila. Okay, one more thing I want to let you know in this step. Whenever you are dying or you are studying this inheritance biology unit, just fully go through that Drosophila concept is very important in the CSIR exam. Drosophila, then in while studying the plant physiology, the Arabidopsis thaliana. This some concepts are very important. They are repeatedly asking this kind of questions. So whenever you are studying this inheritance biology, just well studied this process. The considering of the cross. Okay, this table, the cross and F1 generation, F2 generation. Crosses are two crosses are here. One is gray female X and yellow. One first cross is gray female and yellow male, and next one is yellow female and gray male. They are first generations, or all males are in a gray mode. All males are gray mode. All females are gray. This is the first generation output. All next in the cross 2, all males are yellow. Next, all females are gray. Okay. Then next generation, second, just say F2 generation, they are mentioned 98s are gray female, 45s are yellow females, 49s are gray males. Okay, yellow males 49, gray males are 45 and 49. This ratio they are taken from the they are randomly taken this ratio. Okay, here just they are mentioned they are 200 F2 generation or F2 offsprings are there. The offspring 2 cross, they are asking in a cross 2 question, what is the expected outcome? Okay, here the mention the all grays males, all males are gray in and all females are gray. They are make a second generation. That generation, the most of the males and females are having gray. The most of the females and male, but here they what mention all males are yellow, all females are gray. Okay, we want to understand. So here next generation, the gray female and males also there. One thing first we want to confirm, both male and females are gray, but here not accepted. The males are yellow, the females are gray. So here in this output we should have 4 group: gray female, gray male, yellow female, yellow male. This 4 group we want to select. So this option we want to eliminate first 2 because there is 3 options all alone. So we will get the third and fourth, that is a C and D answer. Next we should confirm what is the probability, because that is the second generation. the second generation mom having a gray, and second generation father, that is the male, having a yellow. So the output in a equal manner, that is not be higher one, because the D answer you can see, the 98 is the male, a gray male, 94 is yellow male. This is not acceptable, because the first generation they are equally having the colors, and so it's not acceptable in the D answer. That is the 52 gray males, 59 yellow males, 58 yellow females, and 51 gray females. This is the answer. Next one is, when two independent pure lines of pi with white flowers are crossed, they mention green, the line of pi's are white flowers are crossed. F1 generation having a purple color, F2 generation obtaining selfing shown both purple and white. So this is the second generation. The ratio is 9:7. The following conclusion made. What is the reason? What is the reason? They asked the reason, that is a PQR. Two different genes. F1 generation they obtained only white, the purple. First generation is a white one, second generation shown a purple one. It's a normal native one is the white one. There's a first generation provide the mixed gene. Okay, they're having a mixed gene. Mutation in a lead to form. If the purple is mutated, that form a white flower. These two genes showed independent assortment. They are individually expressed. And another one is, this is the example for complementary gene action. Action is a process, the complementary gene action is the one important process. This is the reason. Next one, the cell undergoing a meiosis produce 4 daughter cells, 2 of which are aneuploid division only, non-disjunction during second meiotic division. Okay, this is a reason. Next is the following 4 mode of inheritance. One is P, that is X-linked recessive. So Q is X-linked dominant. R is S is autosomal dominant. Okay, this 4 is the option. Which of the above mode or inheritance can be explained with a pedigree diagram shown below? So pedigree diagram, this pedigree diagram that shown the recessive generation. Second one is also the autosomal dominant, because this one is autosomal recessive and one is autosomal dominant. So only they are producing the next generation in a mixed manner. Okay, so answer is C, that the dominant is can be explained in the this inheritance can be explained in the pedigree diagram. Next one is auxotrophic strain, that is the E. coli, that having met minus, bio minus, TR. Okay, these are all the genes which present in the these are all the A having a 5 genes. That gene expression is explained denoted as a plus and minus. Met and bio is minus other than or plus. In a B equalized chain, that met and bio is plus other than others is minus. That is not whether in a 18 hours in liquid complete medium. Then 10 power 8 cells are plated in a minimum medium. The phototropes were observed is frequency 1 into 10 power minus 7 cells. These may have happened by process of genetic recombination between 2 strains or by mutation of this strain. Which of the following to help correct sentence to get a probability of mutation? The C is the answer. Growing strain A and B individually in a liquid medium for 18 hours, then they plating them on a minimal medium. This is the this is the reason for the mutation. Next one is the very easiest question, direct question. The lysogenic cycle is more beneficial to virus. the lysogenic cycle is more beneficial to virus than the lytic cycle under the certain circumstance. What is the lysogenic cycle? Prevent local extinction of host, while still retaining its infection potential. Okay, this is the reason. Next one is also a direct question. The rate of mutation equally from lack negative expression to positive expression that are determining using the medium containing lactose as the only resource and only source of energy. The principal the spontaneity of mutation can be said to be violated. Why this happen means that the reason is B. In the presence of lactate, the rate of mutation from lactate minus to minus lactate plus is increased. That is the presence of lactate, the expression of from the minus to lac plus is increased, but overall rate of mutation is not, that is an oral mutation is not present. These are all the inheritance biology part C questions. I welcome you all to the next class, that is that class tomorrow. We will meet again. If you have any queries, you can post it in the chat box or you can post it in telegram group. In upcoming session from 14, okay, yeah, sorry, that is the upcoming session that is called CSIR crash, and this portion is going to be cover the gate, CSIR, DBT and NCBS, all biological examination at point of view. We are conducting this upcoming session, that is the 60 days crash course. In this 60 days crash course, you can get a better idea and you can learn the topics which going to be asked in a CSIR, gate, DBT and NCBS BCL examinations, because this is the all overall topics we are going to cover. Nearly 120 topics we are selected for discussion. Today I will post it in a telegram group, that I will post the topics in a telegram group, and I will also send the applications also, because that is in Zoom class. If that, because I want to get a direct interaction, so only I make it in a Zoom or Google Meet. So just register on the register for the program and attend, get a better idea for to prepare the CSIR examination. That is very helpful, because November 2nd week